Hey, this is Mr. YS. This is a clip on 1.6 mitosis. We're going to have a look at this video here. Here we see a real cell down a real microscope. And we can see lots of activity. In the center, we can see these structures which look kind of like worms. And you can see that they start to kind of move. And they're moving around the center of the cell. And there's a lot of movement. And then, at one particular point when they're all lined up and everything is ready to go, we see this amazing event where they suddenly get separated and dragged to different sides. And then we get the full splitting of those cells. So this is the process of mitosis, which is the division of two diploid cells, two cells with full sets of chromosomes, to form two genetically identical daughter cells, or daughter nuclei. So this process is happening in every tissue in your body, replacing cells. So why do we need mitosis? What is mitosis for? Have a look at this table and think about what the implications of the figures in this table are. This turnover time is the amount of time that cells, how long do they last for? What's their lifespan? So the implications are, well, we have cells which last for an entire lifetime. The lens cells in the eye. We also have cells in the brain which are going to last for the entire lifespan. And that's okay. They were made once and then they'll just stay there. But the implications from the rest of it is, well, within the small intestine, throughout the lining, with a turnover time of 2 to 4 days, we need to replace those cells. So mitosis is for replacing cells just normally. It could be due to damage. We get a cut, we have to repair ourselves. So we absolutely need mitosis for replacing all of those cells. And it depends on the cell as to how long it lasts, because your central nervous system, spinal cord and your brain, they last the whole time, the whole of your life. So mitosis is there to reproduce cells, and they need to be genetically identical. We have to replace the cell with exactly the same thing, because they have a particular job. There are 220 cell types, and we can't have a muscle cell being replaced by an eye cell. So they need to be exactly the same so that they can carry out the same function. And we have to replace damaged cells or those that become old, so they reach their lifespan. Now, if we take the whole body as an average, then every 7 to 10 years, every cell in the body is replaced, apart from some of those ones that we saw last the whole lifetime. So if you've aged 7 to 10 years, then you are almost entirely a totally new person in terms of the cells that make you up. So on a cellular level, we're replacing everything roughly every 7 to 10 years. But many are much more rapid than that, especially we see cells that are epithelial. So lining cells and also things like skin cells are replaced much more often. The liver cells, for instance, 500 days, and we've got to have this exact duplication. So cells at any one time are always at one point of the cell cycle. They are either growing and copying DNA and getting ready to divide, or they are dividing. And actually this cell division, this is the whole thing, is mitosis. This is only maybe 10% of the time. So they spend 90% of their time just doing what they should be doing, their normal cell function. If it was an eye cell in the retina, it would be receiving light impulses, for instance, and it's going to do that. It's going to do its function. But it will also get ready to divide, to replace the cells that are lost or that are damaged. And so then it's going to be doing the other parts. So we've got mitosis, and then we've got everything else, G1, S, and G2, which are interphase, because inter means between. So if you're not dividing, then you're between divisions, and you're preparing for it. So in some of these different cells, different phases, we need to think about what they would be doing. So what does it need to do? Well, it needs to grow. It needs to copy its DNA, because we're going to divide into two new cells that both need the instructions. If we only had one set of instructions, then the cell wouldn't be able to know what to do once it had divided. And G2, we have similar things happening to G1. So just stop and think about, well, if we're talking about cell growth, what's growing? What does it need to copy or make more of other than DNA? So it's got to copy the instructions, but what other things would it need more of for it to be able to, for one cell to be able to split into two cells which had everything they need to keep functioning? Well, the cell's going to need quite a few things. It's definitely going to need mitochondria, because it's going to need power. So these mitochondria are actually able to divide. They can copy and divide themselves into two. We're going to need lots of ribosomes in order to make more proteins. We're going to need rough endoplasmic reticulum in order to synthesize more proteins. And we're going to need more of every of the cell organelles. So during G1, it starts to copy the cell organelles. And it's also going to need to make lots more enzymes, because enzymes carry out the reactions that happen in a cell. So in G1, we get copying of organelles. We talked about some of the ones it could be, mitochondria. We get more enzymes being made. We're going to need more power for reactions. We've got more ATP being produced. In S phase, it's just copying DNA. So we're going to take all the DNA, all the strands of DNA, and we are going to copy so that we're going to have two strands of DNA. And they're going to be linked together by something called a centromere. So two strands, every strand of DNA is going to become two strands of DNA. And then in G2 phase, we get some further growth, and we also get checking the DNA. So we check that the copying is being done correctly. And if everything's being copied correctly, then we can move into the next stage. Here's our summary. So cells are always in the cell cycle. They're always at some point of it. They're either an interphase, so they're either between divisions, interphase where it's the active period in the life of the cell, where it's going to do its job, do its function in the cell, but also it will then be duplicating organelles, making more mitochondria, making extra cytoplasm, extra enzymes, making more proteins, making more ATP. Then in S phase, this is the synthesis of DNA phase, DNA is replicated. Finally at G2, it's a checkpoint to check everything is done. So check that the copied DNA is okay and we haven't got mutations there. Final metabolic reactions happen and more growth. And then it's ready to divide. It's ready to go into what we saw, which is mitosis. And so this is the role of interphase in mitosis. Well, it's not really it's interphase is before mitosis. So the role of interphase in preparing for mitosis would be better. In S phase of interphase, we talked about the copying of DNA. So see if you can label all the features of this as a recap of DNA structure. If you're doing SL, then these other structures here are HL content. But for HL students, see if you can recap all the different structures. Alright, so if we're in S phase, we are going to copy DNA. Now to copy DNA, we need to have the DNA being loose like this. So this is a loose DNA structure we're seeing here, and maybe it's wrapped around these histone proteins, but it's loose, it's not packaged up. So to copy DNA, it needs to be loose, and this is why when cells are copying DNA, we don't really see anything other than, if we looked in the middle, we would just see a kind of dark nucleus. So in interphase, we don't see chromosomes. If we're seeing this, then we are already in mitosis. We've already coiled the DNA up. So as a quick recap, we've got the simplest level, which is chromatin. Chromatin is our double-stranded DNA around our nucleosomes. So we've got here DNA complexed with histone proteins to form nucleosomes. We've got DNA in between those regions as well, just the DNA in between. And our nucleosome with the 8 histone proteins with the DNA wrapped around. And then we've got our linker protein, which we can call H1, a linker protein. And then that is those are kind of wound up into this 30 nanometer fibre, which goes backwards and forwards, and it makes this 300 nanometer length loops. And these are then kind of compressed and folded down through supercoiling, twisting round and round and round and round. So this is the process of supercoiling DNA, until eventually we get the very condensed structure, which is in a chromosome. And so here what we're seeing, somewhere there's going to be a linking structure, a centromere, and each side is one strand of DNA. So it looks like a fuzzy mess, but in fact, if we kind of drew it as a simple representation, it would be 2 strands of DNA, a single thread, and it might be a very, very long thread all the way along here. It's just one thread, but linking together in the middle is a centromere, and these two at this point, these are identical. It's an identical copy. Supercoiling then is absolutely essential. So supercoiling is winding up that DNA around the histone proteins and then being very tightly associated. So there are enzymes involved in winding them up. And once they get wound up, we can say that they're condensed. So we've gone from kind of a long strand into a very, once it's copied, we would have added in an extra strand. And I'm not going to go the whole way, but it would be 2 strands all the way along. But those 2 strands need to condense into something where they can be easily pulled apart. And so our condensing and supercoiling takes this loose DNA, which once it's copied, it's going to be in this loose form, and it condenses it. So this is condensed DNA, which can now be separated. So at the start of mitosis, it allows the DNA to be arranged. And it's the histones which are allowing for condensation of DNA into a much smaller space and to organise it. And this is going to allow us to separate chromosomes in an ordered manner in mitosis. Let's go through the events of interphase. Looking at this cell here, what can we see? What's in this cell? We can see there is a nucleus, and inside the nucleus we have these lines. So what do these lines represent? Well, this pair here is a pair of chromosomes. And these chromosomes have the same length. And if they have the same length, we can say they have the same structure. Because no chromosome has the same length unless it's the same number chromosome. So let's say for this organism that this is chromosome number 1, and it has 2 chromosomes which are the same length, which have the centromere in the same position, and then if we were to zoom in and look along the length of that chromosome, they would have the same genes in the same locations. But they may have different versions of those genes. So they may have, or will have for some at least, different alleles, the different forms. So these are called, this is one pair of homologous chromosomes. What we're seeing then is 3 pairs of homologous chromosomes. Now when they replicate, they are going to add in new copies of DNA. And they are going to be held together by the centromere. So by the end of S phase, where DNA has replicated, we will have an extra chromosome for all of these. So this is where the language gets a bit confusing. So we said that we had 1 chromosome, and now we've copied it, and yet we can still say that this is a chromosome, and that's confusing. So we're going to call this a double-stranded chromosome to make sure we don't get confused. Before, we had this single-stranded chromosome, and now we have copied, so we've added in an identical strand of DNA. Now, it will have the same genes and the same alleles, because we've just copied it directly, unless there's been a mistake made. So if we were to look at this strand here, and look at the other strand, which it's joined to, if we had big T there, we would have big T there. If we had a little b, it would be little b. All the way along, every gene and every allele will be exactly the same on this double-stranded chromosome. So now, rather than calling each strand a chromosome, although it is, and once they separate, we're going to call them chromosomes again, we are going to call these strands, the strands that make up the double-stranded chromosome, we call each one of these sister, because they're together, they are sister chromatids. So sister chromatids are, when they are separated, back to being a chromosome. And so we could say at the start, rather than double-stranded, when we only have 1, we could call it a single-stranded chromosome. a lot of textbooks and other resources, you won't see this given. So it will be confusing because it is something different. Let's remove all of that and then let's think about what we've got here. So the first question is, well, how many chromosomes does this cell have? And the answer is 1, 2, 3, 4, 5, 6. Now you'll be thinking, why is it not 12? Because I can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 chromosomes if a chromosome is a single strand. And you are right. But now we are thinking about double-stranded chromosomes. So the easy rule is to count by the centromeres. Wherever we've got an X shape, it must be linked by the centromeres. So if I count the centromeres, then I can see I have six double-stranded chromosomes. Now if we ask the question, how many chromatids are in this cell, then the answer is 12 chromatids, because we can count them up, it's each of those fibres. So what about this cell? How many chromosomes are in this cell? Well, the answer again is 6. 1, 2, 3, 4, 5, 6. 6 chromosomes. How many homologous pairs are there? We have 1, 2, 3 homologous pairs, because homologous means the same structure. It means it's the same chromosome with the same genes. And so these are the same length. So we have 6 chromosomes, but they are single-stranded. So this must be a cell that has not yet entered into copying its DNA. It's not yet gone through S phase. When it goes through S phase, it will copy, add in the new sister chromatid to each, and now we're ready for mitosis to happen. Okay, back to our summary. So what are the events of interphase and how could you identify it from a microscope image? Well, it's the replicating DNA, where we have sister chromatids, which are 2 identical DNA molecules held together by a centromere. 2 identical molecules. If we were to look down the genetic code along those, it would be exactly the same unless we've made mistakes. Now I've only drawn in the one side, remembering that this is a DNA helix on both sides. We'd actually have base pairing all the way along these 2 strands. But they would be identical DNA double helixes which are linked by this pointless centromere. So the sister chromatids are what are going to be separated in mitosis to form 2 genetically identical nuclei. Later on, when we pull these apart and put 1 copy into each, then each cell will have the same chromosomes with the same code. How can we identify it? Well, there are no visible chromosomes. So if we zoom in on a cell, and what we see is a kind of just a dark dot, then it's in interphase. As soon as we see any kind of lines, then we're in mitosis. After interphase, we get into mitosis, and the stages of mitosis can either be remembered as just PMAT, or we could use another kind of mnemonic, people, meet, and talk. And so these are going to be our 4 phases. Prophase, pro means before. M for metaphase, and we're going to see a way to remember this is middle. Anaphase, a way to remember that is away. So we'll see chromosomes moving away. And then telophase. And this is where we're going to see 2. You're going to see 2 cells being formed. So PMAT, or people, meet and talk. And notice that what's not there is interphase, because interphase is not a part of mitosis. Interphase comes between in our cell cycle. We will do interphase, then we'll do mitosis, and then it's going to enter interphase again before it gets back round to divide. Stage 1 is prophase. What's happened here? What can we see in this cell? Just stop and think about what you can see. What's happened to DNA which would otherwise be diffuse, be spread out, really spread out? So what's happened here? We can see that the chromosomes have coiled up. We've already had supercoiling because we can see visible X shapes. Once we see this, we know that it's coiled up. So supercoiling has happened. We can say there's been condensation. We've had the DNA condenses, and now we can see these X shapes. We can see that there's starting to be more gaps in the nucleus. I don't think this is trying to show pores. It's trying to show that the nuclear envelope is breaking down, and that's happening towards the end of prophase. The other thing that we can't really see from here is that we have the chromosomes starting to move towards some kind of middle point. So movement of the chromosomes towards the middle point. And if we think about the length of the DNA to supercoil, the length of the DNA has decreased. Here's our summary. In prophase, the chromosomes shorten. They become visible as double-stranded chromosomes. They do that by condensing, by supercoiling, by winding up. And they begin to move towards the equator. And the equator is an imaginary line down the centre of the cell where the chromosomes are going to line up so that they can pull towards each side. In late prophase, we get the nuclear membrane or envelope breaking down. And that's important because then, if it doesn't break down, these chromosomes wouldn't be able to move out to the sides of the cells, to move out of the nucleus. Second phase of PMAT is metaphase. So what can we see happening here? We can see that the chromosomes have moved to the equator. And because they're all lined up, we say that they have lined up. They've lined up on the equator, and we can see that these fibres, these are spindle fibres, or spindle microtubules, which have grown out from the centrioles, which are at the poles of the cell. So from each pole of the cell, spindle microtubules have grown out, they've connected, and actually this is happening in late prophase, to push the chromosomes into the right position. Some remaining microtubules will now attach, and then the cell is ready for the next phase. So what's happened in metaphase is during metaphase, and they're double-stranded chromosomes, all the chromosomes are lined up at the equator separately. The chromosomes attach to spindle microtubules and they attach at their centromere. One important point here is that each of the chromosomes, if this was chromosome number 1, there's another chromosome number 1, and they line up separately in mitosis along the equator. And a spindle microtubule grows out from each pole and attaches to opposite sides. The thing it attaches to is actually a kinetochore, but that's not really part of IB. So we can say they attach to the different sides of the centromere. Each chromosome will attach to a spindle fibre, a microtubule, coming from the poles, coming from the centrioles. So these are two centrioles at the two different poles. The microtubules will grow out and they will attach to those chromosomes at each side of the centromere. And the way to remember this from PMAT is that M is for middle, because the chromosomes are lined up in the middle. Our next stage from PMAT is anaphase. Anaphase we can think of away. So what we see here is that each chromosome that are lined up, each double-stranded chromosome that are lined up along the cell equator and had attached to a spindle microtubule at both sides of the centromere, these now contract, the spindle microtubules contract, and that gives a pulling force. The centromere, the structure in the middle, the centromere divides to allow each of those strands to go their own way. And so if we remember when we had those two strands, it's a double-stranded chromosome, but each one of these was a sister chromatid. So in anaphase, sister chromatids are pulled apart. By the contraction of the spindle microtubules and the division of the centromeres, and they are being pulled to opposite poles. Here's our summary. So in anaphase, we can remember it as the away phase. It's where they move away from each other, the chromatids. So at the start of anaphase, the centromeres divide. And this phrase is a key one. The centromeres divide, and this separates sister chromatids. The separated sister chromatids, once they have separated, and this is again a confusing bit of terminology, so once the sister chromatids have been pulled apart, this was a double-stranded chromosome, we could just say was a chromosome. Once it's been pulled apart, we then have, so the sister chromatids are pulled apart. We only know them as sister chromatids when it's a double-stranded molecule. Sister chromatids are pulled apart, but once they've been pulled apart, we can now go back to calling that a chromosome. So the chromosomes, the sister chromatids separate, and now the chromosomes are pulled to opposite poles. But this is a single-stranded chromosome. And that's happening via contraction of the microtubules. The final phase of PMAT is T for telophase. So what can we see in telophase? We can see that the X-shaped structures have started to disappear. We formed the X-shaped structures, the double-stranded chromosomes, through condensation. So the opposite of that, if we decondense, if the DNA unwinds, now we get this looser form of DNA. So in telophase, we can see that the DNA is starting to decondense and unwind. Chromosomes are no longer visible. Certainly not as the double-stranded. And we're starting to get reformation of nuclear membranes. So the nuclear membranes are starting to reform, and our chromosomes are at opposite poles. After which, we then need to cut down the middle. So telophase goes with cytokinesis. Cyto for cytoplasm and kinesis for the idea of cutting the cytoplasm into two. Here's the summary. So in telophase and cytokinesis, the nuclear membranes start to form around each set of chromosomes. And we have division of the cytoplasm and formation of two new cells at the end of cytokinesis. We also have the idea that DNA decondenses and chromosomes are no longer visible. In animal cells, the splitting of the cytoplasm happens with a contractile ring. So a ring is a microfilament ring, and what it will do is it will cut through the cytoplasm. It starts to contract and it creates what's called a cleavage furrow, and it keeps on contracting until the membranes reform, and now we have two separate cell membranes. And a very high level detail would be that these contracting proteins are actin and myosin. And we'll see that they are used actually in muscle contraction. Whereas in plants, instead of having a contractile ring, there is something called a cell plate, and vesicles which contain substances move towards the equator. They fuse with some tubular structures in the cell plate. So we get these vesicles moving, and they deposit in this gap. They deposit cellulose and pectin, which make up the cell wall. So we don't go into a lot of detail, but yeah, vesicles move fusing with tube-like structures which are at the centre in the cell plate and depositing cellulose and pectin, which creates the new cell wall and forms the new cell membrane, which would be inside. Here's our summary, and this is very much at a 7 level to be describing all of this. So in animal cells, we have a cleavage furrow formed by a ring of contracting protein, which is actin and myosin. Whereas in plant cells, we have a cleavage furrow causing a cleavage furrow to form. And the cleavage furrow is this dip on either side that's caused by this ring of protein that starts to get smaller and smaller. Whereas in plants, we get vesicles moved to the equator at the cell plate, fusing with tubular structures, we get 2 membranes formed across, and we have pectins and cellulose deposited, forming 2 new cell walls.